Hello and welcome to another video on genetic for schools. Now in this video we will learn about the concept of test cross. Now every organism has wide variety of characteristics or traits and each trait can take various forms. For example, height, and the different forms can be tall or short or something like a medium sized organism. Similarly, fruits can also take different forms based on, let us say, size or the amount of sweetness present in them. So there are a wide variety of traits that an organism has, be it a plant or an animal or any other unicellular or multicellular organism. Now, geneticists and breeders are interested to know or determine the genotype of an organism. And the technique that they employ to determine the genotype of an organism is known as a test cross. Certainly, Punnett square is used, but in order to determine physically what is the genotype of the organism that I have, is known as a test cross. Now, for example, if the geneticist intends to know about this plant up here in the image and he intends to determine whether this is a dominating, whether a particular characteristic is a dominating characteristic or a recessive characteristic, and if the organism is exhibiting a dominant trait, is it number one, is it homozygous dominant, or number two, is it heterozygous dominant? But before we proceed further, I have got these three terms that I intend to discuss here, even though I've discussed this in some previous video, but not everybody might have watched it. So let's just discuss these three terms and then later proceed with the content of the test cross. So first thing is, or the first content is homozygous dominant. Now what is this condition? Well, this is a condition in which the allelic partners of the allelic pair are same or identical as shown here by the letter capital T, and let us assume that this allelic pair governs the height of the plant. Now capital T, well, the capital letter always represents the dominating trait, and let us assume that this capital T leads to a tall plant. And because both the partners of the allelic pair are identical, we call this condition as homozygous dominant. Now there is another condition which is T, capital T and small t, which again has a dominating allelic partner and a recessive allelic partner. Now because both the partners are non-identical, they are not identical, we call them as heterozygotes or heterozygous condition. Now in this heterozygous condition, it is still a dominating, because of the presence of this dominating allele, the plant will still be tall, and that's why we call this as a heterozygous dominant condition. Now we have the third condition, which is a recessive condition. Well, the recessive trait is only expressed in the homozygous state. I'm sorry, I'm taking a different letter. Let's use small t. Well, if the plant has to be short or let us assume dwarf, then it can only be dwarf if it has a pair of recessive alleles. In the presence of a dominating allele, the recessive allele cannot exert its influence. Gene may be active, may be producing a protein, but it cannot exert its effect. The effect of a recessive allele is only exerted when both the partners in the allelic pair are recessive. So, recessive traits are only expressed in the homozygous condition. But is there something as a recessive heterozygous recessive condition? Can there be something as a heterozygous recessive? Well, let us see. So if I write it like small t and capital T, you see this is a heterozygous condition, but there is a dominating allele, and the dominating allele will not allow the recessive allele to basically exhibit its properties. And so this heterozygous recessive is nothing. There is nothing as a heterozygous recessive. If you look at this carefully, it is nothing but heterozygous dominating condition. And anyways, the way to always write an allele is the dominating allele is written first and then the recessive allele. So I hope you've understood the three terms: homozygous dominant, heterozygous dominant, and the recessive. There is nothing as a heterozygous recessive, and recessive traits are only expressed when both the pairs or allelic partners are recessive. Now let us proceed further after this discussion towards test cross. Now in a test cross, why does a breeder conduct a test cross? A breeder conducts a test cross to determine two things. And let me just rub it and make my board just a bit clean. And you have to give me just a moment. And here we are. Now I've got a little space. All right. So a breeder, if he intends to know whether the plant is dominant or whether the plant or his organism of choice is exhibiting a recessive trait, the best way to do is to have pure lines. And even to conduct a test cross, we require recessive, a recessive pure line. So if the breeder intends to determine whether the organism is dominant or recessive, that is easy. The person can basically compare this particular plant, forget about whether it is dominating or not, to a pure line which is recessive. So it is easy to determine to distinguish between a dominating and a uh, recessive allelic pair carrying partner, right? But if the geneticist or the breeder intends to know, is my plant homozygous dominant or heterozygous dominant, that is the real difficulty. Why is it so? Let us have a look. So let us say an organism or the same plant is tall, right? This is tall. So why can this be tall? It can be tall because of the presence of the homozygous dominating allelic pair, capital T, or it can be tall because of the presence of a heterozygous dominating condition, which is capital T, small t. Now, by looking at this plant, you cannot say whether this plant is homozygous dominant or heterozygous dominant. Certainly, by looking at the plant, you can say whether this has a dominating trait or a recessive trait. See, if this plant is dwarf, let's say this was just here, right? This plant was just this height, after achieving maturity. Then you could have said and referred it, cross-referenced it with a pure line of a recessive trait. We could have easily determined or distinguished between a dominating and a recessive plant. But by looking at a tall plant, you cannot say whether it is a homozygous dominant or a heterozygous dominant trait carrying plant. How do we determine that now? Now, in order to determine that, whether the plant is a homozygous dominant, number one, or whether the plant is a heterozygous dominant, we conduct a test cross. And what are the requirements to conduct a test cross? Well, the first requirement is we require a pure line containing a recessive trait. We require a pure line containing a recessive trait, right? Or basically a homozygous recessive, which is the only condition, right? So if I have to find out, let us assume for this particular plant whether this plant has a homozygous dominating allelic pair or a heterozygous dominating allelic pair. So what do? I first of all take a pure line, and the pure line will have the genotype small t small t. So this is what I require. I should have it. So once I have a recessive trait carrying allelic carrying plant, I cross it with the candidate, with the plant that I am interested in, that I want to inquire about. So I want to find out about this particular plant, so I would require this plant. So what I do is I will cross my candidate plant with the pure line, which is the recessive. Now there can be three conditions: either my candidate is homozygous dominant or my candidate is heterozygous dominant. So let me assume that my candidate is homozygous dominant and I'm crossing with a recessive trait. So let's fill the first table for these two assumptions. So let me just write capital T here, because these are the gametes. They represent the genotype of the gametes, and I'm writing the gametes for the recessive trait onto the sides, and this is small t, and let's just fill up the Punnett square. So we get capital T small t, we get capital T small t, we get capital T small t, and we get capital T small t, which is basically 100% tall plants will be seen as the phenotype. And basically this is the heterozygous, this is the outcome of the heterozygous condition. If my plant is homozygous dominant, then I should get 100% tall phenotype. But what if my plant is a heterozygous dominant? So let us see what happens. Now heterozygous dominant is capital T small t, and what am I going to cross my plant with? I'm crossing it with a plant that has recessive traits. So I take the recessive traits onto the sides, and now let us fill the Punnett square for the second case, right? So for the second case, what do I get? I get capital T small t, I get small t, I get capital T, small t, and I get small t, small t. So what is the outcome? Now, well, let me just change the marker color and let us have a look at these two boxes. So here, capital T, small t, capital T, small t, I got 50% tall plants, and then I got small t, which means I got 50% dwarf plants. So now, can you see that if my plant was a homozygous dominating plant, upon crossing my plant with a recessive plant, or a plant carrying recessive trait, I'll get 100% tall plants. But if my plant contains a heterozygous dominating trait, then on crossing my plant with a recessive trait or a pure line for recessive trait, what do I get? I get 50% tall and 50% dwarf plants. And this is the way to determine or differentiate between the homozygous dominant and heterozygous dominating conditions. So I hope you have understood. I hope I was successful in delivering across the concept of test cross. Why do we conduct it? We conduct a test cross to differentiate between the genotype of organisms and what type of genotypes, whether my organism is homozygous dominant or whether my organism is heterozygous dominant. I will see you in some other video. Thank you so much.